Hello everyone. What we're going to talk about in this lecture is the structure ultimately of plants. So we'll go through the cell structure, the tissue and organ structure, and ultimately the anatomy of plants. One thing I want to specify before we start is that we'll be talking mostly about angiosperm, so the most evolved and successful type of plants there are now. So we're talking about the flowering plants. So what are we going to touch? As I mentioned before, first we'll quickly discuss the structure of plant cells, particularly comparing them to animal cells. Then we'll talk about the organization of tissues, the type of tissues that you find in plants and how they are organized in some organs. Then we'll go through the anatomy of plants. And finally, we talk about how do plants keep their constant growth, which makes them very different from animals. We stop growing, plants keep growing. How do they sustain that type of growth? In the first part, I will focus on the first three issues, and then in the second part, I'll finish up talking about growth. So, plant cells. Plant cells are eukaryotes. Okay, plants are eukaryotes, so like animal cells, they have different organelles inside. They have a nucleus and so on, different things, mitochondria and so on. However, they have also some unique features. What are these unique features? There are three main differences. First thing, unlike animal cells, they have plastids. What are plastids? Plastids in reality are organelles, part of the same family. We always talk about chloroplasts. Chloroplasts are only one type of plastid, the most famous type of plastids. But in reality, there are other organelles still with double membrane, similar to chloroplasts, but which have different functions, such as myeloplasts, for example, are similar to chloroplasts. They have two membranes, but they do not have thylakoids. And they're specialized in the storage of amylose and amylopectin, storage of starch. So there are more than chloroplasts in there. So all plant cells would have plastids. Whether they are photosynthetic, they would have chloroplast. If they are not, they may have amyloplast. They may have chromoplast, other types. Together with that, they have also what we call a central vacuole. This is a vesicle, which can take quite a bit in terms of structure in the cells. Can take anything between 30 to 40%, all the way to 90% of the whole cells. And this vacuole here is filled with fluid, mostly water. And what it does, it actually compensates, producing what is called the turgor pressure, compensate for the pressure coming from the wall, and therefore helps keeping the cell, keeping the structure of the cell all together. And together with that, third property of plant cells, which differentiates them from animal cells, is the presence of a cell wall. Now, all plant cells would have a primary cell wall. Some of them would have also a secondary cell wall, which we'll talk about in the next slide. When we talk about the primary cell wall, what we're talking about is a semi-rigid structure. So it's rigid but still overall flexible, which is made by cellulose fibers, which are embedded within a matrix of hemicellulose. Hemicellulose is very different, despite the name is a polysaccharide, different from cellulose. Is made by more than one type of monosaccharides and is also branched. So slightly different than cellulose, or quite different than cellulose. So cellulose is embedded in a matrix of hemicellulose and other proteins, forming again this semi-rigid structure. Now these represent, obviously give strength to the cells, but also represent a barrier between the plasma membrane and what's inside the cell and the external environment. And how do cells overcome that, particularly when it comes to connect with each other and communicate with each other or exchange material, is by forming direct connection between the cytoplasms, which are referred to as plasmodesmata, which are mediated by proteins that basically connect, go through the cell membranes and the cell wall through the next cell wall, cell membrane, forming direct connection between cytoplasm of two neighbor cells. As I mentioned before, all cells have a primary cell wall. Some plant cells have also a secondary cell wall. This is on top of the primary cell wall, and its form is a stronger type of cell wall. Okay, And the reason why it's stronger is because among the it has a different structure. First of all, the orientation, for example, of cellulose fiber is different, is done so that they form a stronger mesh overall. And also it's fortified because of the presence of these polymer or phenyl polypropanoids. If you remember when we were talking about the step in evolution of plants, we talk about how cell acquire UV resistance by learning how to produce phenyl polypropanoids, and then they acquire the capacity of making their structure stronger by learning how to polymerize these phenyl polypropanoids, forming lignin. This is the structure of lignin right here. And so this is deposited within these matrix here, ultimo cellulose and so on, hemicellulose and so on, making these secondary wall very thick and much stronger than the primary cell wall. Not only that, but this creates also a waterproof barrier, and that's the reason why you find this type of secondary cell wall also in some cells that are responsible for the movement of fluids throughout the plants, as when we talk about xylem, we'll see later on. Now, this is an example of two cell types that actually do belong to the same type of tissue, which is referred to as ground tissue. They're both so part of the ground tissue. So these are parenchymal cells, and these are sclerenchymal cells. These cells have only a primary wall. You can see a very thin wall here, which allows them to basically take several different type of shapes. They can be even square at a certain point, round, small, and so on. These cells instead, they have a thickened secondary cell wall here, which is typical. When you look at slides, you see usually it has a reddish type of stain, and as you can see, these basically creates a much more even type of shape in these type of cells. Moving on, so we talked about plant cells, the difference between them and animal cells, and the type of cell walls that they have. Now let's move on to what cells form. So we'll talk about tissues and organs. So first of all, by tissue, what we mean is group of specific cells and the substances that are found between them that are unified ultimately by a same specific function. Okay. For example, we can talk about vascular tissue. These are cells that are working together with the goal of moving material across the plant. We can talk about epidermal cells or epidermis as a tissue. We're talking about cells that work together to form a layer that can separate, if you want, the outside from the inside of the plant. Now, tissue can get together and form organs. Okay, so organs are defined as body structure. They're made by two or more type of tissue. One example you see in plants are leaves, where you can find ground tissue, particularly parenchyma, combined with epidermis and vascular tissue, all working together, forming one organ which has a definite form of the typical form of leaves, and the function which is to be perform photosynthesis and exchange gases. So what are the type of tissues that you can find in plants? Well, the type of tissues are very simple overall. There are only three types of tissues, very different than what you find in animals. Plants are actually overall extremely simple, which is probably one of the reasons why they've been also so successful overall. So they have three types of basic tissues, which are ground tissues. They're very simple, but again, they consist of different types. We'll see, we'll talk about parenchyma, collenchyma, and sclerenchyma, which have different functions altogether. Second type is vascular tissues, which ultimately they all are responsible for movement of fluids, whether they are just water and minerals or fluids enriched with organic products of photosynthesis. Still, they're all tissue. They're still vascular tissues. They still move this material around. And the third type, another very simple one, is dermal tissue, okay, which has only the function ultimately to protect the plant surface from the external environment. Let's start with the ground tissue. Now, in reality, there are three types of ground tissues. There is parenchyma, collenchyma, and sclerenchyma. This picture shows you the difference between them. I already showed you a picture actually of parenchymal cells and sclerenchymal cells when I was trying to point to you the difference between cells with a single primary cell wall and those with a secondary cell wall, as you can see here. So let's go through the details of these three different types of ground tissue. So when it comes to parenchyma, this is made by parenchymal cells that form what is called a soft primary tissue. It's called soft because indeed you can easily squeeze it and form it in different ways. They're classified that have only a primary cell wall, which is very thin. You can see in this picture right here and here. Now, in reality, there are different types of parenchymal cells, and the different types reflect also the different functions that they have in the overall plant structure. Here you have two examples. You have parenchymal cells that are found, for example, in the roots, which have the function to store starch. So they have these plastids here, which are filled with amylose and amylopectin. These are a second type that you find in leaves. These are parenchymal cells that specialize on performing photosynthesis, and therefore, instead of having glycogen, so plastid with amylose and amylopectin, my apologies, they are filled with chloroplast, okay, so that they can be extremely efficient at absorbing light and so on. Second type of ground tissue is collenchyma. This is made by collenchymal cells, which at this point start having a thicker cell wall, but it's uneven. It's still a primary cell wall, but it's thicker than in parenchymal cells, but is not evenly distributed around there. So they can be semi-rigid, these type of cells. Okay. Their function is ultimately to produce some flexible type of support. And you see an example here. These are collenchymal cells, which you find in these disgusting things, which is called celery. I hate it. I'm not a big fan of it. But if you think about when you bite into, or if you take some celery into your hands, you can see how you have this long structure, which overall is fairly strong, but still flexible. It's not completely rigid, right? And this flexibility, the strength and flexibility is given indeed by this bundle of long cells, which are collenchymal cells, which ultimately form these bundles and gives it that crunchiness and at the same time strength overall in there. So they are long cell organized again in bundles because of their fairly thick cell wall, primary cell wall. They can give strength, but it makes structure still overall flexible. This is different from what the third type of ground tissue, which is sclerenchyma, does. Sclerenchyma is formed by sclerenchymal cells, which are of different types, and these cells, unlike parenchyma and collenchyma, at this point they have also a secondary cell wall, which is actually fairly thick. It's typical when you look at them, you see them this very thick, even overall red type of secondary cell wall. Is based on the staining that you get this red color. We'll see when we go to the lab and we do some plant histology. Now, because of the presence of the secondary cell wall, at this point they can form fairly rigid type of structures. Okay, structures such as, for example, the one that you find in the shell of nuts, like this one. Look at walnuts. This structure is formed by fiber and sclerenchymal cells. Same thing when you look at vascular bundles here. Now, vascular bundles are those group of vascular tissues, so xylem and phloem, which have the function of moving liquid through the body, all right, or plant body. Now, per se, this cell could actually collapse or they could curve easily, particularly when you have fairly long stems or roots. And so these structures are fortified indeed by the presence of sclerenchyma cells around. So these are sclerenchyma cells that you find here. These bundles here, this bundle right here, and this is the vascular tissue in between. So the presence of sclerenchyma cell with their secondary wall makes this structure much stronger so that the plants can sustain their growth vertically too. Now there are in reality two types of sclerenchyma. I mentioned already both, but there are two types. So you have sclerades. Sclerades are bunches of a few cells, okay, few sclerenchyma cells. They're not organized in long stretches. In this case, you would talk about fibers. So what you find around vascular bundles, in vascular bundles, are actually fibers. What you find instead in nuts, in the nut shells, is actually sclerates. Sclerates, you can find them also spread within some fruits. For example, when you bite into a pear, you get those very annoying grains sometimes. You feel it. Kids particularly don't like them. You have that crunchiness that inside the pulp of a pear. That's exactly caused by sclerates, and that is actually done trying to repel some sort of enemies from actually ingesting these type of fruits. So we talked about the first type of tissue. Ground tissue consists of parenchyma, collenchyma, and sclerenchyma, tissues that have different function, which we discussed. The second type of tissue you find in plants is called vascular tissue. This is that tissue that has the function of moving fluids around the plants. There are two types of vascular tissue, referred to as xylem. Xylem has the function to move water and the dissolved minerals basically from the roots throughout the whole plants, and it moves these liquids in a unidirectional way from the bottom to the top. Whereas phloem is the second type of vascular tissue. Instead can perform bidirectional type of movement, so in both directions, and it has the function of moving organic, the product of photosynthesis, ultimately the sugars that come not really directly from photosynthesis. If you remember when we talked about photosynthesis, we were talking about how it ends with glyceraldehyde 3-phosphate, but then outside of photosynthesis, glyceraldehyde 3-phosphate can be converted into different sugar and organic products, which then can be moved throughout the plants in a bidirectional way through exactly the phloem. So let's focus on the xylem first. So the xylem, it has already a secondary cell wall, which is lignified and therefore waterproof. That's essential to try to keep the water within the as much as possible within the xylem itself. In reality, it consists of thin and large tubes like this one, fairly long, made by fairly long elongated cells, which actually are dead. So these cells that are produced, they elongate and then they die. But because of the presence of lignin and a secondary cell wall, they do not collapse on themselves. They stay connected with each other. Now there are two types of cells and therefore xylem systems. There are tracheids. In this case, the cells are narrower, and also when the connection between them is tapered, so it's not perfectly straight. It's tapered like this. Now these cells, they have pits in between them and also sideways, so they allow for the movement of the fluid also laterally here. The second type of xylem that you find is referred to as vessel members. These are dead cells too, but unlike tracheids, they are larger. Okay, they're larger, it's more evolved. We mentioned that when we're talking about evolution of plants. They're larger, they still have pits that allow for the movement of material sideways, but when it comes to the connection between different type of vessel members, first of all, they're a bit shorter, and the connection is not tapered, but it's actually basically perpendicular to the wall of the vessel members here. And so there are pits that allow for the movement of the fluid more directly in a vertical way. The second type of vascular tissue is phloem. As we mentioned before, it's in charge of the bidirectional movement of organic material within the plants. In this case, this is not formed by dead cells, but is formed by living cells, which however have lost their nucleus, so they are enucleated. So these cells, they form, they elongate, and then they lose their nucleus. That's similar to what happens, for example, to your red blood cells. Your red blood cells don't have the nucleus anymore, and they last about 30 days within your bloodstream. In this case, the cells, they are kept alive, however, by cells which are outside of them, which are referred to as companion cells. These cells still have a nucleus, so they can still perform all the function and move all the material within the the phloem cells here. Now, what they form are called sieve tube members. So these elongated structures, which are connected through sieve plates here, which have pores, which allows for the material to move in both directions. You can see when you take a cross section, so by cross section what I mean, if you cut the tissue, let's say you're looking at a stem of a plant and you cut it perpendicular to the main length, this is how it will look if you focus on the phloem. You can see larger cells, which are the sieve plates or the sieve members here, connected with the sieve plates. Okay, you can see the sieve plates here with the pores. And then on the side, you can see these smaller cells here, which are the companion cells. Now, both phloem and xylem, they do not work separately, but they work together in combination referred to as vascular bundles. Okay, vascular bundles that can have different properties in different type of plants, but which are always supported by additional cell type or additional tissue, the ground tissue referred to as sclerenchyma, which we mentioned before, which is this type of tissue around here with a secondary cell wall, which strengthens overall the vascular bundles. Now, this is a vascular bundle you can see in the stem, so in a cross section of a stem of corn, for example, and this is the one you would see in a cross section of buttercup, of a root of buttercup here. These, if you look, the vascular bundle here has both xylem, has a secondary cell wall which stains as red. So in the xylem here you have the tracheids, which are larger, tracheids, which are the smaller type of vessels. You see them right here. And then you have larger vessels here, which are indeed what we call the vessels. Okay, so you can see both types there. And then the xylem is embedded within the phloem, which is with stained blue. And here you can see the components, the sieve members, these larger cells with the companion cells beside them, which keeps them alive, even though they are enucleated. Okay, and together with that, as I mentioned before, there is the sclerenchyma on the run. Here you can see on the root similar things. So these large components here are the vessels of the xylem. Then you have smaller components here, which are the tracheids, and then you have the phloem here. Together with that, you have the pericycle, the endoderm, other structures here. Okay, we'll talk about that when we talk about the structure of the roots, and we'll see all of these when we go through the histology plant lab together. Last type of tissue you find in plants is the dermal tissue. Okay, and these forms what we call the epidermis, which ultimately has the function to protect the plant. Okay, so they forms a surface on top of the stem, of the leaves, of the roots, in order to protect these structures from the external environment and control what moves in and out. Okay, now usually it's covered by a cuticle, a waxy cuticle, which is nothing else than a layer of lipids or waxes, which basically protects from water loss, particularly. Oftentimes, in leaves, there are stomata, which are openings made by specialized epidermal cells, which allows for some openings to be open and closed. There are two cells here that swells, and therefore they close the opening or shrink, and they open the opening and allow for the exchange of gases. Usually they are found on the bottom of leaf surfaces. Sometimes you may also observe some specialization. We'll see in a second when we'll talk about the leaves. You can see that some of these cells are specialized for example, root hair. These would have some irritant inside it or other structure we'll see in a second. Last part I'll discuss in this first part of the talk is the anatomy of the plant, and we'll see the differences between two particular groups of angiosperms, which are the monocots and the dicots. So in general, when we look at the plant anatomy, again, we focus basically only on angiosperm here. But in general, a plant structure can be divided into two parts. There is the part that is exposed above the ground, is referred to as the shoot system. Okay, this has different function. We'll discuss them in the next slide. And then there is the part which is above ground, which is referred to the root system. In reality, I don't know if you recall, but in the previous lectures, I mentioned how some specific structure originating from the shoot system, which is called the rhizome, like the ginger is an example, or potatoes are part of a rhizome as well, are tubers which are part of a rhizome. They're actually coming from the shoot system, but unlike this definition, they're found below ground. So this is a generic definition or separation between shoot system with root system, but when we talk about rhizome, we're talking about some sort of exception. We'll see also how some roots, the adventitious roots, in reality originate from the shoot system and they can be exposed above ground. So there are some exceptions to that. Okay. Now, both structure here, they sustain, they keep growing throughout their life. That we'll talk about that in the second part. And the type of growth that they have is referred to as dendritic growth because it produces that typical structure which we find in trees or in dendrites, those neurons. Do you remember the parts of the neurons called the dendrites? We'll talk about that in the second half of the semester, which have these ever-splitting structure. They keep splitting, forming this typical dendritic form here. Now, the shoot system. Well, what does it consist of? Well, it's all that part above ground. Let's keep this definition, which consists of stems. The one you see here, this is the main stem. These are the lateral stems. It consists of the leaves. There are buds present here, which are responsible for growth. And then you have flowers and eventually fruits, which then later on, the flowers turning into fruits. Now, what is the function of them? Well, there are two main functions. One is photosynthetics. Being above ground, being exposed to the light, the shoot system has the function of absorbing the light and therefore producing the food necessary for the plant to grow and stay alive. And the second thing is reproductory. The stems, the lateral stem here forming the flowers, clearly they have the responsibilities of producing the pollen, attracting plants animal to the flowers to get the pollen spread, or just exposing the pollen to the air gets the pollen spread and then developing the fruit, spreading the seeds. Now, because of these, the shoot system can be divided into two parts. There is a vegetative shoot system, which is non-reproductive. Okay, it could be basically the stems with all the leaves there. So the part that is not involved in the reproduction. And then you have the reproductive shoot system, which consists instead of the stems with the flowers and eventually the fruits later on. Now, other words I want to use here so you can see how there is a main stem and then you have lateral stems that originate from area referred to as nodes, and the space between these nodes is referred to as the internode. You'll see when we talk about monocots how there is specific tissue here which is responsible for the growth in between the nodes. Now, we talked about the flowers, we talked about the fruit when we're talking about the evolution of angiosperms. Here I focus only basically on the leaves and the difference between monocots and dicots later on in terms of stem. But when it comes to the leaves, what we're talking about is those structures that are specialized in the shoot system to absorb the light, to maximize the amount of light that gets absorbed and also to maximize the gas exchange. So the release of oxygen coming from photosynthesis, and the absorption of carbon dioxide, which is necessary for the formation of organic molecules. Now, this is performed, so there are two parts that you can find on the leaves, what is referred to as the blade. This is actually where photosynthesis takes place. And then the blade can be connected to the stem either by a petiole. There are some specific plants referred to as eudicots. We'll talk about it later on. There is this non-photosynthetic structure here, referred to as the petiole. Whereas in other plants, referred to as monocots, the blade actually directly connects to the main stem through that part referred to as the sheath. Now, how are leaves organized inside? These are organs, so they're combination of different tissues. What are the tissues? Well, you have an external epidermis that you find in all leaves. It's a single layer of epidermal cells, which are usually covered by a waxy layer, the cuticle, minimize water loss through that. And on the bottom, this layer has some epidermal cells which have specializes for the control exchange of gases, okay, they forming the stomata that can open or close. We talked about that when we talked about particularly the C4 and C3 cycles in plants. Within the epidermal tissue here, there is ground tissue, particularly formed by parenchymal cells. Okay. Now, these parenchymal cells form what is called the mesophyll. All of these is referred to as the mesophyll. And there are two types. There is the palisade mesophyll. This is formed by parenchymal cells which are packed with chloroplast, and they have the function is towards the top of the leaf, that the function to absorb as much light as possible. Okay, so the function of the palisade mesophyll made by photosynthetic parenchymal cells is to perform photosynthesis. Whereas the second type of mesophyll, which is referred to as the spongy mesophyll, the name says it all, the cells are not tightly connected with each other or packed together, but they have a lot of room in between them. And the reason is that the spongy mesophyll here, which again is still a certain type of parenchyma, so ground tissue, are specialized in moving the gases in and out, exchanging the gas, allowing the gas to come in or being released. Okay. Together with that, there is a third type of tissue here, which is vascular tissue organized in vascular bundles, which then would have xylem, phloem, and other cell types, so sclerenchymal cells, which are part of the ground tissue, just to going through the naming again. Now, some of the leaves have actually specialized in different plants, forming different structures. Here you have some example here. You have tendrils. These are found, for example, in these are in invasive plants, such as ivy, for example. Their structure, they're responsible to actually attach the plant to pre-existing structures, such as walls or other plants. This is another example of specialization I mentioned before. This is stinging hair. These are actually structures which are filled with irritant fluid. So when an animal approaches this plant, you can find them also on stem, by the way, not only on leaves. These animal would actually break the stinging hairs, and the contact with the fluid will make the animal react, potentially even deadly react. And then you have some leaves that have specialized actually to become some sort of digestive organ. And as you can see in these tropical plants called Venus flytrap, where you have leaves that have been specialized on closing, actually, and by doing that they can actually trap insects. Not only that, they don't only trap the insects so that they can't leave, but they also produce enzymes which allows the digestion, and then they can absorb the product of digestion through the surface. So there are different variations of the different leaves that you can find in different plants. Now, moving to the second part. So we talk about the shoot system. Now, let's talk about the root system. This is the system that usually grows below ground with some exception. We'll see in a second. The functions are different. The root system originated first of all to anchor the plants to the ground, but most importantly to allow for the plant to absorb water and minerals, sometimes forming mycorrhizas with pre-existing fungi. The other function that roots have is to store carbohydrate. Just think about carrots, for example. It's an example of a root where there's a lot of carbohydrates stored there that gives that sweet taste. to the carrots. Now, in terms of structure, based on the way they grow into the ground, there are two types of roots. We have tap roots and fibrous roots. What do we talk about? When we talk about tap roots, we're talking about roots that tend to grow vertically toward deeper, deeper, and deeper in the ground. So they would have a main root, which grows very deep, and very short side roots. Carrots are an example of that. Here they've been removed. When you buy them, the side roots have been removed. But again, this will be the main root that would have grown deep into the ground. Several plants like lily, most eudicots actually have tap roots. Okay, they don't make them as great plants to actually hold the ground itself. They don't really hold the ground. So if you are planning to build or retain the dirt from falling, let's say into the sidewalk, this is not the type of plants you want to put in there. What you want to put is plants that they have the second type of roots, which are referred to as fibrous roots. Fibrous roots, instead, they grow more laterally in all directions. You see it in grasses, for example. And so they are specialized particularly to absorb water more than store carbohydrates. Okay, and they're found mostly in monocots, again, such as grasses and so on. Now, there are other type of roots that are referred to as adventitious roots. In reality, these roots do not originate from the root system, but they originated from the stem of plants, oftentimes under stress circumstances, such as lack of water, strong lack of water, so the plants need more and more water, so the stems start producing additional roots, which then will get in contact with the water. One example you see is the one, typical one, you see in Angkor Wat. I don't know if you have been there. It's an amazing place. I highly recommend you. It's in Cambodia. It's an old lost type of temple that you have in there, and there all the temples were actually completely covered by these plants of the genus Ficus, which actually indeed produce all this structure here, which are nothing else than adventitious type of roots. Now, let's finish up this first part by talking about the difference between these two main groups of angiosperm plants. Again, angiosperm plants are the flowering plants, the most evolved plants, which can be divided into different groups. In the old days, they were divided into dicots, with dicots including basically what now we call eudicots, and a small group of primate dicots, which are like magnolias, so it would have been a paraphyletic type of group, and monocots, okay, like palms, bamboo trees, and so on. Nowadays, the classical division is between monocots and eudicots, since the primitive dicots represent just a small group of plants, and oftentimes they are included to eudicots, to the point that eudicots and dicots are actually oftentimes used as the same word. So just to give you some numbers, about 3/4 of the angiosperm nowadays will be eudicots, where the monocots are about 25% of them. So what are the differences between these plants in terms of anatomy and structures? Well, there are different differences. First of all, the name in itself says that monocots have only one cotyledon, which is that primitive seed leaves that you find in the seed. If you remember, the early embryo, the early sporophyte found in the seed has already some primitive leaves in there. So if you have only one, the plants will be a monocot. If you have two, will be a eudicot. Okay, you would have two of them, two cotyledons, as they call. Now you see some example of plants that belong to monocots: grasses, lily, cattails, corn, rice, and so on. You see them right here. Now there are lots of differences between these plants, which allow them to be recognized. We already talked about the cotyledons here. Now, in terms of flowers too, in general, monocots, they have 3 petals or multiple of 3, whereas eudicots would have 4 or 5 petals found in there. Other big difference is how the vascular bundles are organized within the leaves. In monocots, the vascular bundles are parallel to each other. So, and you see, particularly if you take a cross section of a leaf, you will see them nearly separate. Instead, in eudicots, the vascular bundles are forming this interconnected network. And so if you take a cross section, you don't really see very well organized type of vascular bundle. We'll see that in the lab. Together with that, there are also big differences in terms of how the vascular bundles are organized in the stems and in the roots. Other types, also the root system. So usually tap roots are found mostly in eudicots, and whereas the fibrous roots are found in monocots. But let's look now at the structure of the vascular bundles in the stem of eudicots and monocots and in the roots. What are the differences? Sorry, another difference which I mentioned before, I'm just reminding you before I move to the structure of the vascular bundles, is how the leaves attach to the main stem. So in eudicots, you have a petiole there, known for photosynthetic structures, whereas in monocots, the blade itself basically continues until it attaches to the main stem, with the part connecting to the stem referred to as the sheath. In terms of organization of the vascular bundles in monocots and eudicots or dicots stems, what is the difference? Well, if you look here, this is how a cross section of a stem of a eudicot plant will look like, and you'll see one in the lab. And this is how a cross section looks in a monocot plant. These are the vascular bundles. As you can see, in eudicots, the vascular bundles are nicely organized in a ring structure here. Ring structure which then determine the separation between what is called the cortex, this tissue here, and the pith, this tissue inside. So you can clearly see a pith inside these plants. Whereas in monocots, the vascular bundles are spread all throughout the stem. You don't have a ring structure, but each one of these is a vascular bundle right there. There are differences in the way also the xylem and the phloem are organized in there, but particularly they are spread throughout the entire, within the parenchyma here inside the stem. This is a little bit different what you see in the roots, where you actually have that in eudicots, you have what is called the stele. The stele is the vascular bundle basically, plus this vascular cylinder that you have in there, plus the pericycle, which is a layer of cells, undifferentiated cells basically, and the endodermis. The endodermis here, which is this circle right above pericycle here. So you have a single one right in the middle of the root. That's what you see in the eudicots. Okay, and so the cortex is very, very thick, very large right here. You don't see a pith in the eudicots. Something instead that you can see in the monocots, where you have that now instead opposite to what you see in the stem, you have that the vascular bundles here are organized within the stele. The stele is this structure right here, the overall thing, in a circular manner. So you have several vascular bundles here, all organized in the circle here. And so they separate what it will be the pith from the cortex right here. You have a clear separation in there. Now, finishing up on this lecture, just using some words which are important later on when we'll talk about growth, to understand the growth, the lateral growth of roots. When we look at the roots of plants, so this will be the root of a eudicot, so with a still here, singular basically vascular bundle there, and not pith present in there. So when we look at this, we can identify different parts here. So you have, first of all, an exodermis, is the external epidermis that you see here, and then you have an endodermis here, which is what define ultimately where the stele is. Underneath, you have a layer of undifferentiated cell. We'll see they are referred to as some sort of meristem or meristematic cells, which is referred to as the pericycle. Okay, it's right below the endodermis. And then you have the xylem and the phloem, sorry, the xylem here and the phloem here in blue, nicely organized and separate from each other. You can see when we talk about growth, the role of the pericycle. So these undifferentiated meristematic tissue here in the lateral growth of the plants. You can find these cell, by the way, also in monocot roots here. So you have the endodermis, and then the pericycle is present right below here. So allowing these plants to form lateral roots as well. We stop here. In the second part, we talk about specifically the growth of plants.